My name is Abigail, or mostly just go by Abby Gerhold, and I am an assistant professor, which means that I am pre-tenure. I started here in 2018, and I am well ensconced in the molecular side of the biology department, so my lab studies cell division. I actually did my undergraduate degree in English literature, and that was because I enjoyed reading, I enjoyed writing, I enjoyed a lot of the sort of philosophical discussions that one has as part of the sort of humanities. But then when I graduated, I wasn't sure what I was going to do professionally, job-wise. I sort of bopped around in a bunch of different types of positions and was having a hard time finding something that I enjoyed doing on a day-to-day basis. And I got a lab position, somewhat fortuitously and somewhat just by brute force. And I just loved research. I really loved the day-to-day aspects of research. I loved the process of discovery. I'm very into microscopy, what we can sort of see these days with microscopes, sort of what's going on inside living cells, the level of sort of resolution and the ability to see these things happen in real time. That gets me very excited. So I could spend most of the day at the microscope, given the choice. The ability to see some of the stuff that we can now, I think we're really kind of at the cusp of understanding much better how cells work. Because honestly, it's always been sort of amazing to me that they do work, right? When you think about the complexity of tasks that they undertake, and yet when it boils down to it, it's all sort of biochemical reactions, and sort of just the fact that it works still sort of astonishes me. So being able to watch some of that in real time, I think, is really cool. We mostly work with a model nematode called C. elegans, and the reason why we work with C. elegans is because it's a great system for live cell imaging. And so we can do live cell imaging of cell divisions during embryogenesis, so as the embryo develops. And we can also do live cell imaging of adult stem cells in a living intact worm. And so what that lets us do is ask how mitosis happens under more varied circumstances. So we know a lot about how mitosis happens in cells grown in culture. We have a lot of mechanistic understanding about how cells build a spindle and they line up their chromosomes and they segregate their chromosomes, and this is all very important for maintaining genome stability. But what we know a lot less about is sort of how that happens within the complex physiology of a living animal or within a complex sort of tissue organization. And so our research kind of aims to tackle that question by asking, if you start playing around with the animal's physiology, maybe by changing growth factor signaling pathways or changing its environmental conditions, what happens to the fidelity of mitosis, and how does the animal sort of maintain high fidelity mitoses under those circumstances?